comparing at our knowledge about the physiological and the biochemistry of abiotic stresses. The abiotic stress tolerance varies with the nature of the stress, salinity, heat, frost, and so on. That is according to the developmental stage.
Dr. Yasser, no voice. Dr. Yasser, you are here?
For example, some plant at late stages and so on.
Dr. Yasser, excuse me. Dr. Yasser?
Yeah, genetic mapping approaches that, such as untitled analysis, we have to. I'm here. Are you hearing me?
I think your voice have some problems. You need to raise your voice a little.
Are you hearing me right now?
Now his voice is okay, and you can use full screen for your BBT.
We try to manage that, but it doesn't manage, Dr. Muhammad.
Okay, go ahead. Thank you. I'm sorry to disturb you. Let's
try it right now. Is it working or?
I think
الصوت بيقطع بس يا دكتور ياسر. الصوت بيقطع شوية بس.
Okay. Is it right now with the full screen or not?
Not.
Okay.
Okay. Go ahead.
Yeah, the first question was, though the same genomic regions control the variation at different growth stages, or different genomic regions are controlling the variation of abiotic plant response to abiotic stresses. The second question was, though the same genetic regions control the plant response to different abiotic stresses, for example, drought, salinity, and heat, or different genomic regions are controlling the response to the different types of abiotic stresses. The research outline, is it okay with is it okay with the PPT or?
Okay, go to the next slides.
Okay, you can see the next slides, or?
No, you are now in the third slides.
Okay.
Introduction.
Okay.
Yes, now it's shift. Okay, go ahead.
Okay, now it's slide number 5. Okay,
yeah.
Let's continue like this. Okay, this is the research outline. We have published 2 research papers concerning with drought and at early stage, and 2 publications about drought at later stage. 1 publication at about salinity stress at early stage, and 1 publication about the heat stress at late stage. The workflow was to phenotype the collection and to genotype the same collection using SNP or SSR marker, and then to do QTL or GWAS analysis or single marker analysis, and then to make a gene annotation to identify the candidate genes. Let's start with the first stage. We can title it genetic control of drought stress at early stages. Here we have published 2 papers. The first one entitled Genetic Basis of Drought Tolerance During Seed Germination in Barley. It has been published 2018 in detailed genetic analysis for identifying QTLs associated with drought tolerance at seed germination and the seedling stage in barley. Let's to start with the first one. I will not go much deep in the technical details. But before going to GWAS analysis, 8%, reaching at 95% is very high heritability. And then we have made the correlation analysis, and from the correlation analysis, we observed that the correlation between the germination-related traits was always with the seedling-related traits. For here, the correlation was negative between germination percentage and root length, and so on, except the correlation between germination base and root length, it was positive, but not significant. After the GWAS analysis, we found that the most important associated significant SNPs have been identified on chromosomes number 1, 2, and 5. The SNPs on chromosome 1H were associated with seedling development traits. The QTLs or SNPs on chromosome 2H were associated with germination or seedling related traits. The SNPs on chromosome number 5 were associated with germination traits. After the gene annotation, we have identified several genes from this table. I'll put much more focus on some genes. For example, the first gene, that the orthologous of these genes are positive regulators of drought tolerance in Arabidopsis and rice. And in our case, it was associated with root length under drought. As I mentioned before, for some traits, the correlation was positive between seed germination and seed development traits. If we look here, this gene that is highlighted in the blue color, this gene controls the variation of germination base. That is a germination-related trait. Also, at the same time, it controls the variation in root length. Might be this gene causes this positive correlation. Or in other words, this gene controls more than one trait at a time. And they are very valuable genes if we would like to select for improving more than one trait at the same time. The conclusion of this paper, the early stage can be subdivided into two different stages: seed germination and seedling development. Additionally, different genomic regions control seed germination and seed development. Genes on chromosomes 1 and 5 can be targeted to improve germination percentage. Genes on chromosome 1 and 2 can be used to improve the seedling development traits such as root length and shoot length. The best genotypes in this collection were originated from Europe, European Union, East Asia, and America. Then coming to the next publication. In this publication, we have used an Egyptian spring barley collection. The first collection was a worldwide collection, and this collection is Egyptian collection. When we make the correlation analysis, we get the same results. The correlation between the seed germination related traits was always negative with the seedling development traits. This confirms our hypothesis that the early growth stage can be subdivided into two sub-stages: seed germination is a stage, and seed development is another stage. If we look here about the number of significant SNPs per chromosome, we can find that chromosome 1 has the highest number, and the chromosome 2 and chromosome 5 like the first collection. Additionally, chromosome 3 and the chromosome 6 harboring high significant number of SNPs, and this is specific for the Egyptian collection. If we look here, we can see that chromosome 1 harbors the SNPs for root length, while chromosome 2 has a combination of SNPs that control germination-related traits and SNPs that control root length, while chromosome 5 harbors SNPs for germination traits, and this quite similar to the first population. While chromosome 3H here harboring SNPs for germination traits, and chromosome 3 and 6 in case of the Egyptian collection has valuable alleles compared at the worldwide collection that we have seen earlier. Yeah, with gene annotation, we have identified several genes distributed in several chromosomes. I'll put more focus in some genes, especially that coding for proteins that already known to have something to do under abiotic stress, such as the genes that coded catalase 1 enzyme or catalase 3 heat check protein, late embryogenesis. All of these proteins are known as responsive molecules for abiotic stresses. The genes on chromosome 3H were specific for fresh weight, under control, and under drought, while the genes on chromosome 6 are specific for germination-related traits. Therefore, if we would like to improve the fresh weight, we will select for the alleles on chromosome 3H, and if we would like to improve the germination percentage, we will select for the markers on chromosomes 6H. The final conclusion is, if we compare our results that we found in these two populations, we can see that in the worldwide collection, chromosomes 1, 2, and 5 harbor the highest number of SNPs, the most effective SNPs. The most effective SNPs, we meant that they are controlling more than one trait at a time. Therefore, these markers can be targeted to improve these traits together. In the Egyptian collection, we also found that chromosome 1, 2, and 5 still have something to do. Additionally, chromosome 3 and 6 have valuable alleles. When we compare the number of QTLs that we have detected for a trait such as root length, that is very important trait under drought stress, we found that there are 33 QTLs, and this is a large number. If we compare our results with the worldwide collection, in the worldwide collection we have identified 36 QTLs. Abdul Ghani et al. Have identified 34 QTLs for seed germination related traits in another collection. Nas et al. Have identified 19 QTLs for root length. It means the Egyptian collection has several variable alleles, even though the number of genotypes was very low. It's about 60 accessions, while the worldwide collection was 220 accessions. It means the Egyptian collection has sufficient genetic diversity. The conclusion of this paper, again, we came to a conclusion that different genomic regions control seed germination and seedling development. Genes on chromosome 3 and 6 can be used to improve fresh weight. We have identified the most drought tolerant genotype, and the genotype PNPY-T15 showed the best drought tolerance, and this we can recommend that this genotype can be used in the breeding program as a donor for the drought alleles. Then we come to the next step of our work that dealt with genetic control of drought stress at late stage. We have finished the seed germination, seedling stage at 2 different collections. With the same 2 collections, we will continue working, but at flowering, fruiting, or maturity stage. The first paper has been published in Genes, entitled Natural Variation, and covers the Candida number and the green yield under drought stress. The second paper, entitled Marker Trait Association for Grain Weight of Spring Barley in Well-Watered and Drought Environment. For the first paper, we have used the worldwide collection. For the second paper, we have used the Egyptian collection. Yeah, also the correlation analysis. In the correlation analysis, we can find that the traits in the same stage, for example, the heading related traits showed high and significant correlation, even under control or under drought. When we made the GWAS analysis, we found that the most effective SNPs have been identified in chromosomes 2H and 3H. And if we look here at these arrows, we can find that there are a large number of SNPs that co-localized at the same position, or they are linked. And if we look here in the linkage analysis, we can see that in the chromosome 3H, we have highly linked SNPs. This means there are SNPs representing the same QTL, and they will be co-inherited, and they will be transferred from generation to generation together. Therefore, we have focused only on these SNPs that I have shown previously, that are co-localized or they are highly linked, to identify the genes that are associated with these SNPs. We have identified several genes, but I'll put more focus only on 3 genes. One of them controls the number of grains per spike exclusively under drought, and the second gene on chromosome 2 controls the variation of number of grains per spike under control and also under drought. The third gene controls the variation of 10 different traits at a time, and this one is a very important gene. Then we have looked for the gene expression profile of these genes. To simplify that, we have developed another graph for these 3 genes. The first one with the orange color, if we look here, the expression pattern of this gene is a bit noisy. It goes up and down, and the change is very high, while the variation in the other 2 genes is a bit steady state. And if we come back to the table, we can see that this gene is adaptive. It means the gene expression of this gene is induced under drought stress, while these 2 genes are constitutive. And it has been demonstrated that the gene expression of the adaptive gene is noisy. It means there are extreme changes. It goes up and down very strongly, while the constitutive genes, if we look in the gene that is colored in the blue color, the gene expression variation is a bit steady. There is no high and low peaks. The conclusion of this paper is chromosome 2H and the chromosome 3H harbors the most effective genes that are related with number of grains per spike in barley. The gene expression pattern of the adaptive genes is more noisy than the constitutive ones. The most important gene that is reside in chromosome 3H that controls the variation of 10 traits at a time. Therefore, this gene can be targeted for selection to improve these traits together. Yeah, the next paper is entitled Marker Trait Association for Green Weight of Spring Barley in Well-Watered and Drought Environment. This experiment was a real field experiment. We have conducted it on the Egyptian collection that includes 16 spring barley genotypes, and we focused on the green yield per spike and green weight with some other yield-related traits. We have calculated some tolerance and susceptibility parameters. This figure shows the distribution of the southern kernel weight drought susceptibility index and the green yield per spike at the two different locations where the experiment has been conducted. The most important thing in this paper that we have identified a single marker that controls the variation of both seed germination traits and yield traits. We have combined or analyzed the markers with the seed germination related traits that we have discussed earlier in the Egyptian collection, and we have found that a single marker controls the variation of southern kernel weight as well as fresh weight at the germination or seedling stage, and this only the single marker that we have found to have something to do under two different growth stages, at seed germination as well as at maturity or southern green yield per spike. Then we have identified the most drought tolerant genotypes. If we look here, we can find 4 genotypes showed the highest performance or outstanding performance at the two different locations, even under control or under drought. And these, among them is this genotypes PNPY T15 that was the most drought tolerant under the most drought tolerant at seed germination and seedling. This means that this genotype is showed a steady performance at different growth stages. And this table shows us the selection parameters. We have considered some selection parameter if the genotypes, if the genotype occurs 4 or 5 times in these parameters, we consider it as drought tolerant. If we can see here, PNPT15 was among the best genotype in 5 traits, and CSYT28 showed or was among the best genotypes in 4 traits, and so on. This is the marker that we have identified to be associated with the variation in southern kernel weight at the maturity stage, and this marker also controls the variation of fresh weight at seedling stage it means this marker can be used to select at early stages based on the performance of the genotypes or the yield of the genotypes of fresh weight. Yeah, we have asked ourselves a question. Is there any common genomic region that controls the variation of drought at seed germination or and at late growth stages? When we compare or combine the SNPs that we have identified at seed germination with the SNPs that we have identified at late stages, we didn't find any common SNPs. The final conclusion is that in the given collection, I mean here the worldwide collection, there is no common SNPs between the seed germination and the seedling stage and the yield stage. In other words, different genomic regions or genes control drought stress at different developmental stages. Then we come to a different type of abiotic stress, salinity stress. At seed germination, we have used the worldwide collection for this experiment. Also, in the correlation analysis, we observed that under control, the here in front of the arrows, the correlation between the seed germination traits and the seedling development traits was non-significant. But under salinity, the seed germination traits and the seedling development traits showed positive correlation. This is only under salinity. Under drought, the correlation between the seed germination traits and the seedling development traits was always negative. Therefore, we can see that the plant response differs under drought about under salinity, even at the same stage. Yeah, this is the ANOVA table, and we can see that the heritability was very high. It was always higher than 90. It was always higher than 90.
Dr. Yasser,
yes.
Dr. Yasser, time is up, Dr. Yasser. I hope we can finish earlier, okay? We can give you just 1 or 2 minutes maximum.
Okay, I'll try to be, it's something like 3 or 4 minutes. I'll try to be on time. Okay. This is a list of the genes that we have identified. If we look here, we can find that the most of the Candida genes have been identified at chromosome 5. Okay. Among these genes, we can see that they are responsible for or acting as, or they are coding proteins that acting as magnesium transporter, potassium channel, and squamose promoter binding proteins. If we look here, we can find that each gene of these genes controlling more than one trait, and they are controlling seed germination traits and seedling development traits. This explains this positive and significant correlation under salinity stress compared at under drought. Here we can see that chromosome 1 and chromosome 5 harbored the most or large number of clusters of SNPs, and the conclusion of these papers is under salinity, the germination traits are more related to seed development traits than under drought. Chromosome 5H harbors the highest number of significant SNPs as well as the highest number of Candida genes and it's the first time to localize the squamosal promoter binding like protein in barley in this study. And when we compare the SNPs for drought with the SNPs for salinity, we didn't find any common SNPs between the SNPs that we have identified under drought with the SNPs that we have identified under salinity at the seed germination stage. The last paper is about the effect of heat stress at reproductive stage in barley. In this experiment or in this paper, we have employed the Egyptian collection, and this paper entitled Investigation of Heat-Induced Changes in the Grain Yield and Grain Metabolites with Molecular Insights on the Candida Genes in barley. Yeah, as I have mentioned before, in the Egyptian collection, under drought stress in the field study, we found that the green yield berry spike was a very effective trait. Therefore, based on these traits, we have classified the collection into 4 groups. We have analyzed some physiological parameters such as protein content, proline content, and the carbohydrates, and so on. If we can see here, some traits have been increased, such as chlorophyll content, amino acid, protein, and carbohydrates. The remaining traits decreased. When we conduct the principal component analysis, we found that green yield per spike and yield per plot and the starch were the most effective traits, or they are the traits responsible for the variation within the collection, and this is the ratio or the contribution of each trait in the variation within this collection. We can see that green yield per spike and starch, water content, and protein were the most effective traits. When we subdivide the collection into 4 groups, we can see Group A, where the reduction in the trait was less than 10%, all the parameters increased in this group, especially the protein content and starch, while in the remaining groups, these parameters decreased. When we conduct the gene annotation, we have identified the most important or candidate genes on chromosome 1H and the chromosome 4H. These genes coding transmembrane transporter activity proteins, and this is very important because, as we know, at flowering and grain filling, the resources should be translocated from stem and from root and from leaves to be reserved in the grains. This means these genes are very valuable, especially at these growth stages.
Dr. Yasser, time is up, Dr. Yasser. We take more than 5 minutes. Okay.
This is the final.
I'm so sorry.
Yeah.
So thank you for your nice presentation, and now the time for discussion. And I am sorry, we will take just one only question because there is no time. Okay, so for the audience, if anyone have question, please raise your hand, and Dr. Ahmed Salam will be unmute you, or you can type your question in the chat box. Okay, I will take just only one question because of time. Thank you.
Welcome.
Okay, Dr. Ahmed Salam, you see any question in chat box? Dr. Ahmed? Okay, I can see.
Yes, we have.
Okay,
go
ahead.
Question.
Okay.
In the first, I want to thank Dr. Yasser for the nice presentation. I'm very happy to attend the informative presentation. I have some questions. My question is, can the seedling vigor cause a difference in germination percentage from one concentration to another?
Yes. Yes, for the concentrations that we have used before, yeah, identifying the optimum concentration, we always conducting a preliminary experiment to identify the optimum concentration. Then we start with the pilot experiment and we phenotype the whole collection. And we found that when we used concentrations like 15% of polyethylene glycol, these show a mild effect. When we used 20%, we find large variation. When we used 25% of polyethylene glycol, the variation increased or the response differs. It means that there is a different response with the concentration. When we increase the concentration, the variation becomes larger. Therefore, we just expect that if we have used different concentration, might be we can identify different QTLs or different genomic regions related to each concentration.
Okay, Dr. Ahmed Salam. Thank you, Dr. Yasser, for your informative presentation. And now we go to the second speaker, Professor Mark Tester. Are you here, Professor Mark?
Yes, I'm here. Thank you very much.
Thank you. Okay, please, Dr. Yasser, close your share.
Okay.
Give a chance for Professor Mark to share his presentation.
I did so.
Okay.
So how do I do that?
Yes, you have open share try. This is like arrows near to the mic.
I have the microphone. Yes, open share tray.
Yes.
Good. And
you get
now.
It? Yes.
I hit that.
Yes.
I think I need control or something.
Yes.
Doesn't do.
You have blue bar. This is have mic and camera, and this is square with have inside of this square have arrows. Click in this arrows. Yes.
Yes, and that's the share tray. But now what do I do?
Dr. Ahmed, can you fix this issue? Dr. Ahmed Salam.
Yes. Hi, Dr. Mark. How are you? So you have a small, you have an arrow beside the mic. You just click on it.
Arrow. I can click on it to close it and open the share tray. But then when I open the share tray, all I have is a black bar across the bottom of my screen.
You should have an option with open share for screen one, or you don't have like this. Okay. I can't see even. I
open the share tray, and all that happens is a black bar appears, but it's just black.
Shall I say something?
Yes.
Yes.
Yeah, Professor Mark. If you click on this this arrow, it will open you a window. Then you can select to go to your PC, and then you will find the presentation.
Yeah, it's not opening anything.
Oh.
All it's doing is raising in the black.
Yes, it should open two windows. For example, here in my case,
no, it
opened a window. I can see you and I can see Dr. Muhammad, and there's a right window. If I click in this window, I'll go directly to my PC and share my presentation.
No, it's not. It's not showing anything. Oh dear.
Okay, so I don't.
Dr. Ahmed, please, can you switch this presentation to Dr. Yasser, and he can show Dr. Mark how to, for example, about how to share.
Okay, yes. I can share it here. Okay, I will do this. Yes. Okay.
Yes.
So, do you see my screen?
Yes.
Okay, so I think now you can see yourself and Dr. Muhammad.
Yes.
So here this is like a small an arrow. Just you click on it, and then you will find the point that if I click on it, it
Okay.
will stop the
I
sharing.
need to get control. I've just pressed request control. That's why nothing's
Okay.
happening.
Okay, you can do
So
this.
can somebody give me control, please?
I did.
Okay. That's not worked. Damn.
Yeah, because you are in my screen. Okay, so please
Oh.
just. So could you please cancel
Somebody give me control.
Could you cancel the control, please?
Oh. Okay. Stop control, right? I've done that.
Okay, so here this is an arrow, and you just click on it, and then you press on the screen. So if you pressed on the screen, then you will get your presentation appear to us. So
Okay, look, I've pressed the share. I've pressed this.
I should
this open. I've pressed open share tray.
Okay.
And as I've said many times, okay, now I'm going to try again. Open share tray. All that happens is a black bar appears across the screen. I'm trying to hold the mouse down on stuff. I'm holding the mouse on open share tray. Hold the mouse. Nothing is happening.
Dr. Ahmad, I think Dr. Mark can send it to you his presentation, and you show from your PC, and just Dr. Mike give connect about this.
700. It's 700 megabytes. I'd have to.
Oh my God.
Oh, it's really weird.
Yeah.